So, good morning, everyone. Welcome to WESTalks. Uh, as you know, just to give a brief intro on IC Impacts and Future Waters. So, IC Impacts is a Canadian Canada-India Research Center of Excellence, which works on a lot of collaborative research in the area of infrastructure, health, and water. And UBC Future Waters is a research center or an excellence cluster based out of UBC, which works on the intersection of water research, environmental work, and policy and governance. So just to give you a brief intro, who we are in WESTalks. So basically it's been organized by 4 students, Abhishek, Jaskaran from McGill, myself and Carl, who are at UBC, and Feria from IC Impacts' office. So welcome to week 14. Today we have Dan he'll be giving a presentation on DBPs from drinking and recycled waters. So thank you so much, Dan, for supporting our WESTalks and being a part of it. It's really wonderful to have you here today.
Sure.
Just to give a brief update or to all our attendees that all our WESTalks are available on IC Impacts' YouTube page. So if you are interested and if you have missed any of the talks, you can go to the YouTube page and access all these talks. And as every week, the new initiative that we have started to inform all the attendees what's happening this week. So for the curious people, tomorrow, October 10th is World Mental Health Day. So it's on mental health awareness. Also 12th of October, which is next Monday, it's Canadian Thanksgiving, as well as the Native American Day. So just make sure that you celebrate it. Now, getting to the main point, today's speaker, Dan, Dr. Daniel McCurry is an assistant professor of civil and environmental engineering at the University of Southern California. Dr. McCurry completed his PhD from Stanford in 2016 with a minor in chemistry and a major in environmental engineering. Prior to Stanford, he earned an MS from Yale in civil engineering and worked at the US EPA Office of Research and Development. He received the Rose Hills Foundation Research Fellowship in 2019 and NSF Career Award in 2020. So Dr. McCurry's research focuses on protecting public health by improving the long-term safety of engineered water sources. So he does a lot of work with the reuse facility in Los Angeles. So really excited to look forward to his talk. He also applies the tools of environmental organic chemistry and analytical chemistry to water quality challenges related to chemical and UV disinfection of wastewater and drinking water, with a particular emphasis on identifying mechanisms and precursors to DBPs. So with this, I'll pass it on to you, Dan, and really look forward to listening to your talk.
All right, thanks, Vihar. I think I need to be co-host or something to share screen.
Yeah. Just a second.
No worries. Well, while he's doing that, thanks for the invite. And Vihar asked if I would come talk about DBPs kind of in general. So this is mostly not a research talk. So if any of you work in this area, you may find some of this a little bit boring. My apologies for that. But hopefully it's informative or educational for most of you to some degree. Excuse me. Let me just get situated and put all your heads in the right place here. Okay, yeah, so I'm going to talk about disinfection byproducts in a fairly broad general sense today, mostly in drinking water, a little bit in recycled wastewater towards the end, which is what I do most of my research on. Even though this isn't a research talk, I put a couple of research plugs in at the end because I can't help myself. But anyway, here we are. So as with any talk about disinfection byproducts, I like to begin by saying I'm not opposed to disinfection. Disinfection is probably the single most important public health intervention that we have done as a species in the last 100 years or so. So this graph, I've replotted some data showing typhoid fever death rates in the US throughout the first half or so of the 20th century, showing that prior to about 1900, it used to be pretty common to get and die from typhoid fever. So about 30 per 100,000 population. If you integrate that over, let's say, a 60-year lifespan, that's about a 2% chance of dying just from typhoid fever, not to mention all the other waterborne illnesses that can kill you. But then beginning in 1908 with the first plant in New Jersey that started disinfecting with chlorine, and this process rapidly spread after that to other plants as they saw how well it was working. You can see that typhoid fever death rates plummet to basically zero by about the middle of the 20th century. And the story is largely the same for other waterborne illnesses. So now it's an extremely rare event in any part of the developed world for somebody to get and especially die from a waterborne illness. You know, it makes like major news when, for instance, somebody in Louisiana gets a brain-eating pathogen that kills 1 or 2 people. That's a major news event versus it used to be, you know, quite common to die of waterborne illness. But nonetheless, the reason I'm here today is because disinfection of water with oxidants, you know, most commonly chlorine, is associated with certain chronic health risks that we ought to minimize. Again, you know, the most important thing that we can do is minimize the acute risk from pathogens, but once that's taken care of, it would behoove us to minimize chronic risk as well. And so the number one thing people think of with disinfection byproducts is bladder cancer. There's epidemiological associations between drinking chlorinated tap water and bladder cancer. So in these epidemiology studies, the epidemiologists will do things like go find some town in Spain where everyone has the same lifestyle and the same diet and everything. Half the town drinks chlorinated water and half the town drinks unchlorinated water, groundwater or something. And then they can compare rates of things like bladder cancer between the two populations, and they see a clear increase in bladder cancer among those consuming chlorinated water. It's also associated with certain birth defects. Just mentioning this to point out that it's not only cancer, there's other chronic health effects, as well as preterm delivery and low birth weight. So these are all motivations for us to figure out what it is in chlorinated water that's causing chronic health problems in populations. And so the source of this risk was first identified in the mid-70s or so. It was simultaneously discovered most famously by J.J. Rook at the Rotterdam Waterworks in the Netherlands, but also at the same time at actually the same US EPA lab I used to work at in Cincinnati, long before I was there, obviously. But in the 70s, they found chloroform in drinking water. And as with any, you know, chlorinated compound, their first assumption was that it was an industrial pollutant. You know, you can get chlorinated solvents and things in your drinking water if somebody upstream dumps it from a factory into the river. But what these chemists in the mid-70s found was that the concentration of chloroform in the water source itself, here plotting the data from Cincinnati, showing basically 0 chloroform in the Ohio River. If you then sample inside the plant, it shoots up to 22 micrograms per liter. And then if you go down the distribution system, allowing the chlorine to react for a few more hours, you get up to 100 micrograms per liter, which would actually exceed current regulations. So this has spawned a whole field that's been going now for, I guess, 46 years on how we can minimize the concentration of disinfection byproducts in drinking water. And also, you know, the area that I work more closely in is to understand the chemistry of their formation such that we can re-engineer water treatment processes to avoid formation of disinfection byproducts. And the sort of central dogma that's emerged over the last 40, 50 years is basically that you have natural organic matter, which is just the, you know, source of organic carbon in most surface waters that comes from things like plants falling in the water and decaying over time through a combination of biological activity and, you know, photochemical reactions, things like that, to form this highly heterogeneous organic precursor material called NOM. And then when you apply chlorine to that, you form byproducts. And there's been greater than 700 of these identified, thanks to the work of analytical chemists like Susan Richardson at the University of South Carolina. And 11 of those are regulated in the US. So the first sort of major topic I'll talk about here about DBPs more generally is regulations and what those look like across a few different places of the world. I'll talk about the US, Canada, the EU, and China. And during talking about regulations of these things, I'll mention some things about how they're formed, which tells us, you know, maybe gives us some hints about how to control their formation. So starting in the US, where I am, the US EPA regulates disinfection byproducts as part of the Safe Drinking Water Act, which is the overall regulatory framework for drinking water in the US. And so they regulate 4 what we call THMs, which is this top row of chemicals, trihalomethane. So this is just a single carbon, a single hydrogen, and 3 halogens, which can be either chlorine or bromine. And then 5 haloacetic acids, which is the bottom row there. So kind of similar idea, you have the same carbon backbone for each of these, and then a series of different halogen substitutions. So these were the first disinfection byproducts discovered, which is probably why they're the main ones that are regulated, both in the US and around the world. THMs were discovered in 1974 as DBPs, and haloacetic acids not long after, I think in the late 70s or so. The reason you may ask yourself why these are the ones that are regulated if there are 700 that have been identified, probably A, because they were discovered first, and B, because they're easy to measure. They tend to occur at relatively high concentrations, so you don't have to get like huge concentration factors or have like a super sensitive mass spec to be able to see these things. You can see them with a plain old GC ECD, which would have been available to researchers in the 70s, no problem. And yeah, they tend to occur at kind of high microgram per liter concentrations versus some of the DBPs that we care about at the frontier of research these days might only occur at the parts per trillion or nanogram per liter level. But they could be more toxic, and so we're still potentially concerned about them. Excuse me. So I said I would say something about formation. So the formation pathways of THMs and HAAs are pretty well worked out at this point. There's a series of pretty well understood organic chemical reactions that I won't bore you with for transforming phenolic organics that tend to make up the backbone of NOM into haloforms like chloroform, the first one here with 3 chlorines on the single carbon. And a lot of the same chemistry applies to HAAs as well, so I won't spend too much time on that. Another one that we have regulated in the US and many other parts of the world is bromate. So bromate is kind of unusual for being an inorganic disinfection byproduct. Most of the 700 I mentioned earlier are organic chemicals. Bromate is inorganic. It's the chemical at the far right here. And I'm sorry, I won't have too many of these complicated chemical schemes in the presentation, but this is one that I quickly stole from this Urs von Guten ozone review from about 20 years ago, showing the basic pathway for bromate formation. Bromate is the compound on the far right here. Can you see my mouse if I'm using it to like wiggle around things? Cool, thank you. So it's BrO3 minus. It's a known carcinogen, and it's primarily formed by ozonation of bromide. So for many water sources and many water treatment plants, this would never be a concern. Ozone is not so commonly used as a primary disinfectant, at least in North America for drinking water. So we don't tend to have big problems with bromate formation because we just don't use ozone that much. Also, to get bromate, you have to have bromide. And we'll talk about bromide occurrence in a little bit, but it's very heterogeneous and not all water sources will have bromide. So there's a good chance if you're a drinking water plant in North America treating surface water, it's fairly unlikely you would have enough bromide in your water source to worry about forming bromate. But nonetheless, this is how you form it. It's a pretty straightforward reaction with ozone. You just keep increasing the oxidation state until you get to the fully oxidized BrO3 minus. This figure also shows an option for bromate control, which is, I think, the most widespread option that plants use these days. So people figured out probably about, I don't know, 20 plus years ago, that on its way from bromide to bromate, at the second oxidation state it hits, which is OBr minus, hypobromite. This is the conjugate base of hyperbromous acid, which looks kind of like the brominated version of bleach, essentially. And so hyperbromous acid can oxidize ammonia to make monobromamine. And so if you add ammonia to the water treatment plant along with the ozone, you'll tend to sort of pick up this bromine in the hyperbromite form before you get all the way to bromide. So that's one example of using the formation chemistry of a disinfection byproduct to design strategies for controlling it during treatment. Last one that we have regulated in the US is chlorite. That's the compound in bold there, ClO2 minus, which is, again, I believe a carcinogen. You can get chlorite in a few different ways, primarily from the use of chlorine dioxide as a disinfectant. So there's been kind of this recurring theme in water treatment and DBP research where somebody will figure out that a particular oxidant starting first with chlorine produces harmful byproducts. Then somebody will try to figure out a different oxidant that can also disinfect water, can also kill viruses and bacteria, but doesn't make harmful byproducts. And it will get touted as like the new silver bullet, the byproduct-free disinfectant. And then it doesn't take very long for somebody to figure out a byproduct that disinfectant also forms. So despite, you know, we have, I don't know, maybe 6 or 7 total disinfectants in the toolbox at this point as environmental engineers, and every single one of them is associated with some kind of byproduct. There is no silver bullet, and I'm convinced that there never will be, because, you know, the very thing that makes chemicals good at disinfectants is that they're reactive. And so if they're reactive, they're going to react with other things as well. Unless you completely remove all the organic precursors, for instance, or you know, distill the water before disinfecting it. I don't think there's any future scenario where it's not a complicated system of trade-offs when choosing a disinfectant, rather than just pick the one that works great every time. Anyway, so chlorine dioxide got a lot of people excited for a while because it's an oxidant, but it is not a halogenating agent. Despite containing chlorine, it doesn't chlorinate things. It just steals an electron from them. So chlorine dioxide is a radical compound. it has an unpaired electron, so it badly wants one more electron, and it will go steal that from the first thing that it can find. Often that first thing might be a virus or a bacterium, which could lead to it dying. But chlorine dioxide can also react with itself through something called in chemistry a disproportionation reaction, where two things at the same oxidation state react, and one of them goes down and one of them goes up in oxidation state. So one of the chlorine dioxides can lose an electron and one of them can gain an electron, and you end up with chlorite as a potential byproduct. Also chlorate, which we'll talk about in a little bit. Also, during reaction with natural organic matter, chlorine dioxide again can steal an electron and just oxidize the NOM, so showing the NOM as a radical cation here. So it's reduced form of, the reduced form of chlorine dioxide is chlorate. So just during the process of reacting with pathogens or NOM, it can form chlorite. Lastly, outside of chlorine dioxide, you can also get chlorite just from storing bleach for too long. So bleach, which is just a solution of free chlorine, which is primarily hypochlorous acid, HOCl, and hypochlorite, its conjugate base, OCl minus in orange there. If you've probably realized this if you work in the lab and you do anything with chlorine, like if you have, you know, a bottle of Sigma Aldrich brand chlorine in your lab fridge and you standardize it from time to time to do a disinfection experiment or any other kind of chlorination experiment, you may have noticed that the concentration of free chlorine in that bottle slowly declines over time. That's why you always have to standardize it. If you ever read a paper where they just say they bought 5% sodium hypochlorite from Sigma Aldrich and then they assumed that that was actually 5% and used it, that's not great, because the concentration, even outside of anything else for it to react with, even outside of photochemical reactions, if it sees light, it'll just naturally degrade over time, again through a disproportionation reaction between hypochlorite and itself, leading to chlorite and chloride, Cl minus. So that's another way you can get chloride. It's just an impurity in bleach solutions. And then I said it lists regulations in other places. So in Canada, you have actually pretty similar regulations. These are, in the US, we call them MCLs, which is maximum contaminant level. In Canada, they call them MACs, which is, I think that's maximum acceptable concentration. Any of you Canadians can correct me if I'm wrong on that. I think that's what that stands for. Yeah, they're broadly pretty similar. So you regulate THMs and HAAs, bromite, chlorite, and chlorite. And then also one other one that we don't have, which is NDMA, nitrosodimethylamine. So that's a compound that I'll talk about here in a little bit. But quickly just wanted to also say for chlorate, I discussed chlorite earlier. Chlorate can form from some of these same reactions with chlorine dioxide. And my apologies, I incorrectly described this as a disproportionation reaction before. This is actually probably more accurately described as a hydrolysis than a disproportionation. And then NDMA is a bit more complicated, and it's something that I've done some research on and we'll talk about briefly towards the end. But the basic idea for how you get NDMA, which is this super, super potent genotoxin, and that's why it's regulated at a much lower level, right? 40 nanograms per liter, that's parts per trillion, as opposed to these, which tend to be in the microgram per liter or part per billion range. It's because it's a much, much more potent genotoxin and likely carcinogen. So NDMA forms principally during chloramination. So chloramination happens when you add chlorine to ammonia containing water, right? So hopefully you can again see this little cursor here. So if you add chlorine, which we approximate as hypochlorous acid, to water that has ammonia in it, so this could be either wastewater in like a wastewater recycling operation, or you can just get ammonia in plain old drinking water. When I was a grad student, we had a pretty broad surface water sampling project, and we got some water from Minnesota at some point that had 0.5 milligram per liter of ammonia in it, which was pretty shocking. And I talked to somebody from the utility there, and they said, it was really interesting that apparently up there, and perhaps you have the same problem in Canada, they can get ammonia deposition on the snow. I guess there's enough gaseous ammonia in the atmosphere from industry or agriculture or something that enough of it can deposit on the snow such that when the snow melts in the spring, they can get a pretty sizable amount of ammonia in their surface water. Anyway, when you add chlorine to ammonia containing water, and you probably remember this from your drinking water treatment class when you learned about breakpoint chlorination, you can form monochloramine, this compound in blue here, which is a weaker oxidant and disinfecting agent than free chlorine, but it still is a disinfectant and is pretty commonly used. But then if you have extra chlorine, if you have a high chlorine to ammonia ratio, you can eventually form dichloramine, this compound in red here. It's structure depicted here with the two chlorines in orange. And that can react with secondary and tertiary amines to form ultimately NDMA. The details of this mechanism are not so important for today's talk, but just know that NDMA comes from chloramines. You can also make it in other pathways as well. You can generate NDMA by ozonation of certain things, but the chloramines are the main source. And then just to speak about the rest of the world a little bit, in the EU, according to their council directive, the regulations are actually kind of minimal, only 100 micrograms per liter of THMs and 10 micrograms per liter of bromate. That bromate regulation is probably more relevant to Europe than North America because they use quite a bit more ozone in Europe than we do. But as far as I'm aware, countries in the EU, I think, are free to set their own regulations. So I think there are independent regulations from this in like Germany, for instance. Switzerland, which is not in the EU, has its own THM standard of 25 micrograms per liter. That's quite strict, actually. That would be pretty tough to get under if you're in North America treating river water, for instance, which tends to be pretty rich in organics. But the Swiss are fortunate that their water source is generally pretty fresh. Like if the water in your river that you're treating used to be snow in the Alps yesterday, it probably doesn't have a whole lot of organics in it. So they're lucky in that regard. China's drinking water regulations, which largely parallel WHO recommendations, are kind of interesting because they regulate different THMs separately. So they have different regulations for chloroform and bromodichloromethane than bromoform and dibromochloromethane. If you ask me, these are probably kind of backwards. They permit a higher concentration of the more brominated species, which tend to be more toxic, as we'll discuss in a little bit. They individually regulate a couple of haloacetic acids, and then the rest of the story is going to sound pretty familiar with bromate, chlorite, chlorate. A few other more obscure DBPs that I don't really have time to talk about. Cyanogen chloride is an interesting one because you can pin almost all of its formation on glycine. If you chlorinate glycine, you can make cyanogen chloride at super high yield. Which is kind of an interesting story that we'll talk about later if there's time. Okay, so there's a whole lot of regulations in the world for different disinfection byproducts. And so the question is, are those regulations doing their job? Are they protecting public health and minimizing chronic health risk of things like getting bladder cancer? And the answer is we just don't really know. And part of the reason for that is because the total sum of byproducts we've identified to date represents less than half of the actual greater pool of disinfection byproducts in drinking water. And this is a story that keeps emerging over and over again across different classes of byproduct compounds. So most DBPs tend to be halogenated because we tend to use halogenating agents like free chlorine to disinfect water. And so this pie chart in the top left, that's like this super classic thing that you'll see in any DBP presentation anywhere, which actually is kind of hard to find. I had to do some digging to find the original source of this pie chart, and it's in a book chapter from Susan Richardson like 8 years ago. I don't know why they didn't just do us all a favor and publish this in Water Research or something, but it's in this kind of obscure book from Springer from 8 years ago. Anyway, point being, you can measure total organic halogens with a TOX assay in water. And if you measure that and then you measure all the individual known halogenated compounds, this isn't all of them here, but this is the bulk of the mass of them, you come up with less than half of the total pie. Same story for nitrosamines. I talked about NDMA earlier, but NDMA is not the only nitrosamine. You can also nitrosate other amines. And so a paper from Bill Mitch's lab about 6 or 7 years ago figured out that NDMA itself is only about 5% of this total pie, and the rest remains unidentified. And then in a brand new paper that just came out, I think just a few weeks ago in ES&T from Emily Marin and David Sedlak's group, they were studying the formation of aldehydes, of carbonyl compounds in recycled wastewater, because they wanted to know what is the organic carbon in the finished product of a wastewater recycling operation made of. So I don't have a lot of time to talk about water reuse, but we spend a lot of money when we're recycling wastewater to get everything out of there that we can. You know, we use reverse osmosis, which is supposed to squeeze out everything besides water molecules. We use a UV advanced oxidation process that's supposed to oxidize everything. And yet there's still dissolved organic carbon in the final effluent of these plants. And so they got interested in what this DOC is, and they managed to attribute almost half of it to various carbonyl compounds like formaldehyde, acetaldehyde, acetone, but still less than half of the overall pie. So this might be a problem, especially because the compounds that have been selected for regulation generally are not the most toxic compounds that are known. So, you know, THMs and HAAs are what the EPA primarily regulates, and many other agencies around the world. But looking at this kind of combined genotoxicity assay from a Susan Richardson paper from 2007, you can see that THMs, halomethanes would just be another word for THMs, and haloacetic acids are among the least toxic compounds known. So what we do know is that brominated and iodinated compounds tend to be among the most toxic versus chlorinated compounds. So in other words, this kind of suggests that the chlorinated compounds like chloroform are doing almost nothing, and really it's the brominated and iodinated compounds on the halogenated side that are the most toxic. Also, we know that nitrogen-containing disinfection byproducts tend to be much more toxic than non-nitrogenous byproducts. So almost all of the research that I've done to date in the disinfection byproduct world has been on nitrogen-containing DBPs because they tend to be much more toxic. So I'll just walk through a little bit of where these three classes of compounds come from, and that'll pretty much wrap us up. Fuhar told me to talk for 20 to 30 minutes, so hopefully I managed to fit inside of that time frame. So the way you get brominated stuff, which might not necessarily be intuitive, if you think about adding chlorine to water, how do you end up with brominated compounds instead of chlorinated compounds? Well, there's some pretty straightforward chemistry that gets you there. So bromide is oxidizable. And so if you add free chlorine, HOCl, to bromide containing water, be it groundwater, surface water, doesn't matter, you can form this compound, hypobromous acid, pretty fast. So this is the second order rate constant for this reaction. At a typical free chlorine dose of like 2 milligrams per liter, this corresponds to a pretty fast half-life of about a quarter of a minute. So this is more or less instantaneous on the time scale of drinking water treatment. And you form HOBr, which is a brominating agent, just like HOCl is a chlorinating agent. And that's how you form brominated DBPs, just from very similar reactions with NOM that chlorine can do. Bromine can do the same types of reactions and actually much faster than chlorine can to make brominated stuff. This kind of begs the question, where does bromide come from? You know, it's in it's in certain drinking water sources, but not all of them. Its distribution is highly heterogeneous. So there's a few sources of bromide. One of them seems, I don't know, kind of absurd, is sea spray. So it turns out that just ocean waves crashing on the shore can cause little tiny aerosols to form, and those aerosols can float aloft for, you know, hundreds or thousands of feet or maybe even miles, and end up in a river or a reservoir or something. So if you're a city planner and you put your town's drinking water reservoir right next to the ocean, you're probably going to get some bromide in there. You can also get it from geology. If you use groundwater and you happen to sort of lose the geologic lottery and your groundwater aquifer contains soluble bromide containing minerals, you can get bromide in the source water that way. And then also a more recently identified source is from hydraulic fracturing. So if hydraulic fracturing operations are allowed to take their flowback water, which is what comes back out of the well and tends to be very, very salty and contain bromide among many other things, if they're allowed to discharge that into wastewater treatment operations, which then discharge into a river, you can get increased bromide that way. And that's been demonstrated with a number of different papers, including this ES&T paper on the bottom right by my old lab mate Kim, to result in a shift from chlorinated to brominated byproducts at even, I think they saw it at dilution ratios as little as 0.1% flowback water in the surface water. You could see a huge impact from that. Excuse me. For iodide, it's a pretty similar story. Iodide tends to occur at much lower concentrations than bromide, but you can still get some of it in source water. And for iodide, that gets oxidized to an iodinating agent, also relatively fast here. So this is well, I don't know, maybe not fast. I guess at a moderate rate with a half-life of about a half hour under typical chlorinating conditions. This tends to go through monochloramine rather than free chlorine. I don't remember if I have a slide on this or not, but if you have free chlorine instead of chloramines, iodide actually gets oxidized even further to iodate, IO3-. So it's really only a problem when you're chloraminating rather than chlorinating. And this iodinating agent, or approximately this iodinating agent, which is why I put an asterisk here, because iodine chemistry is really complicated. A couple papers just came out last year and this year by Mike Rose, who's now a postdoc at University of Maryland, Baltimore County, but did his PhD in Lynn Roberts' group at Johns Hopkins, entirely focusing on iodination chemistry and finding that it's actually quite complicated, usually go through ICL or this protonated, goofy, like hydrolyzed iodine looking thing. But nonetheless, if you chloraminate iodide-containing water, you can form iodinating agents, and just like chlorinating agents and brominating agents, they can react with NOM to form iDBPs, which are quite toxic. So again, here the source of iodide can be natural, just like bromide. Iodide and bromide tend to co-occur to some degree. So you could get it from sea spray, but you could also get it from geology. So this paper in ES&T Letters that came out last year sort of mapped the occurrence of iodide in groundwater all around the US and found that it's highly heterogeneous, and it depends on the geology that you happen to be located over. In some places where you see like big circles or dark circles, you can get high natural iodide concentrations, in other places not so much. You can also have anthropogenic sources of iodide, which can lead to iodinated disinfection byproduct formation. And this is kind of an interesting one. So people did some very clever analytical chemistry detective work recently and found that the source of iodide could actually be from this chemical called iopamidol, which is an X-ray contrast agent. So in hospitals, they will feed this to people or maybe inject it in them, not exactly sure, prior to doing certain types of imaging. So on the right here, this is a scan of somebody's brain, pre and post injecting this, this contrast agent. And after injecting the contrast agent, which I guess is good at capturing X-rays or whichever type of imaging source they're using here, and you can see these 3 brain tumors after adding the contrast agent. So you might notice this contrast agent has 3 iodines on it, right, in the 3 positions around the ring that are available. And so if you photolize that, if you run that through UV light, which is used during certain water treatment operations for disinfection, you can release iodide. So the curves here, they're not exactly labeled great, but the curves that are going down are showing a decrease in iopamidol concentration, and the curves going up showing release of iodide. So you can produce a bunch of free iodide that way. Last thing I'll talk about is I mentioned that nitrogen-containing disinfection byproducts tend to be much more toxic than plain carbon-containing disinfection byproducts. Just again showing this figure in the top right here about that. And so for nitrogenous disinfection byproducts, the source of the nitrogen is kind of an interesting question because there doesn't tend to be a whole lot of dissolved organic nitrogen in just plain NOM, in ordinary surface waters that are pristine or free from anthropogenic impact. So for the most part, nitrogen is going to come from wastewater, or at least in the places that I study, it tends to come from wastewater, whether that be potable reuse of wastewater or de facto, sometimes called unplanned wastewater reuse. Wastewater is full of nitrogen, you know, for reasons you might imagine. And so when you put that into the environment or when you deliberately recycle it, you end up with a bunch of organic nitrogen available to be a disinfection byproduct precursor. I won't talk about potable reuse a lot, especially because I saw in a few weeks you have Bill Mitch coming to talk about specifically DVPs and potable reuse, so I'll leave that mostly to him. I'll just quickly though mention de facto reuse as a source of nitrogen for nitrogen-containing disinfection byproducts as a really, really common phenomenon. When we say de facto reuse, what we mean is that, you know, if you live on a river and then downstream of you there's another city on that same river, you know, you're going to take water out of the river, turn it into drinking water, consume it, use it in homes and businesses, send it to the wastewater treatment plant. They'll treat it, get rid of a lot of the stuff in there, but not everything, and then discharge that back into the same river that you used as your drinking water source, ideally downstream before you get your drinking water. But then somebody is downstream of that. And so when they take that river water and use it as their drinking water, it's going to be, you know, some percentage of it will have been wastewater. So there are classic examples of this all over the world. This one on the left here is the Rhine River in Europe, showing its origin in the mountains in Switzerland, where it's probably pretty pristine, flowing through much of Europe in a bunch of cities, including Strasbourg and Cologne. And so by the time it gets to Cologne, or especially by the time it gets to Rotterdam, there's probably quite a bit of treated wastewater in the Rhine River. Likewise, on the right there, that's the Trinity River in Texas in the US, which is Dallas's drinking water source, but also where Dallas deposits their treated wastewater. Downstream, it becomes Houston's drinking water source, and during dry portions of the year, the Trinity River is 100% treated wastewater effluent, principally from Dallas and Fort Worth. So Houston, whether they care to admit it or not, is practicing potable reuse. And so last thing I'll say here, and I think I'm almost on time, is among all these nitrogen-containing disinfection byproducts, there's 2 that we study quite a bit in my research group. Those would be nitrosamines like NDMA, and then halonitromethanes like chloropicrin on the bottom there. Chloropicrin is a highly potent genotoxin, which was used as a chemical weapon in World War I. And we've spent a lot of time studying how it gets formed during drinking water treatment and wastewater reuse. So just a couple quick words about both of these, and then I'll wrap up. So for NDMA, we published a paper just a couple years ago about how NDMA is formed from chloramines. There had been some debate in the literature for a few years about whether there was any role for monochloramine in the blue here, or whether it was entirely dichloramine that was responsible for NDMA formation. And so we more or less conclusively eliminated a role for monochloramine in this paper a couple of years ago, and the reference is there. I won't talk about it anymore because I said this isn't a research talk. And then the last thing, we've been studying for about 3 years now the origin of halonitromethanes during wastewater recycling. So what my PhD student Lily found in a paper we just published earlier this year in ES&T is that there are certain compounds in wastewater, particularly anything that has an N-methylamine functional group, so a nitrogen with a methyl group hanging off of it. This class of compounds includes exciting things like methamphetamine and MDMA. Any compound in this class, if you ozonate it, which is a common disinfection step during water reuse, you can form this chemical nitromethane, which is a small, highly polar industrial chemical that's also a very strong carbon acid, meaning it can deprotonate. So since it can deprotonate at this carbon here, you form a carbanion, which can easily be chlorinated to chloropicrin or other halonitromethanes. So that's all I'll say about that research. There's the reference for the paper if you're curious. And then I wanted to give a quick shout out for some further reading if you're interested in this topic, a few resources that I have enjoyed reading. On the bottom right there is David Sedlak's book, Water 4.0, which despite the kind of goofy title is a really, really good overview of the past and current practice and I guess the future of water treatment on Earth. And I actually make my sophomores read this in class. He also published one with Urs van Guten, a good perspective paper in Science about 10 years ago now called The Chlorine Dilemma that is a quick like 2-page overview of this topic. Urs van Guten published another perspective piece in ES&T just a couple of years ago about oxidation processes more broadly, so including both disinfection but also micropollutant oxidation, which they do a lot of during wastewater treatment in Switzerland. And then finally, Bill Mitch published a paper just, I think last year with Xingfang Li, or maybe 2 years ago, about where we are on DVPs. So I know there's a lot of information. Happy to hang around and answer as many questions as you have. I should, I guess, quickly thank my research group, even though this isn't a research talk. This is most of us on the roof of my building in Los Angeles. And yeah, I guess thanks for financial support. And on the bottom right there is my lab website and lab Instagram. So that's it, thanks.
Perfect, thank you, Dan. That